No.
老师你
No.
好
Hey, nice to meet you.
nice to meet you. How's your weekend? Been a decade.
Oh, so right now you can speak Chinese a little bit.
I'm speaking Chinese back then, but 一点点.
Okay,
我的
very good.
我的中文不好.
It is actually it's good enough to make me understand.
You great, 老师.
Yeah,
You
yeah,
great. You're
everything is
doing
okay.
great.
Yeah.
Good.
So you can hear me very clearly.
Pardon me?
Can you hear me very
Sure,
clear?
sure. Yes.
Okay, okay.
Should we try share your screen?
Oh, so I need to share my screen on the for the PPT.
Yep.
Oh, I will try. Hmm. Oh, I don't know how to share the PPT. Wait, just
Is this your first time using Zoom meeting?
yeah. I used to Zoom, but I just use the Google Meeting.
Google Meet or other platform. Yeah. Zoom meeting more
Yeah.
familiar in Indonesia.
Oh, but I don't know how to put my PPT.
Oh,
Perhaps I can share the whole screen.
if you don't mind, I will share your PPT you sent to me back then.
Oh, but the file is so big. Is it okay?
The PDF?
Yeah, perhaps I can share the whole screen.
Oh, you can click the share icon in the bottom of your screen.
Oh,
There is
but
share,
I can share the screen. The click is available, not available.
not available. Uh,
Yeah.
Thank you, Lause. Mas Elga boleh minta tolong.
So how can I do next?
Iya, sebentar.
Eh, ini punyanya Profesor Lin bisa bisa share screen atau dibatasi?
Sebentar. Bisa kok.
Bisa ya. You can try loh, She. You can click the share icon in the bottom of
Yeah,
our screen, share, and you can uh, go to the folder you save the files.
I'll try. Okay. This one?
Yeah, that one, this one.
This one?
Yep.
So can you see?
Yeah. Can you slide?
So bigger
Yep,
right
yep.
now?
Nah, like
But
that.
right now I cannot see you.
King Chana, I'm here. It's okay.
Okay, okay. So I just just look the PPT and try to read it.
Oh, if it's more convenient for you, I can share your slides, your PDF slides you sent to me previous days ago.
But it's okay. But it's okay. I just because I just modify the slide a little bit, and I
Oh,
also put a very brief video
mm-hmm.
around 8 seconds. So I think that the file will be the file size will be bigger.
Okay then. So
Hmm.
could you please stop share your screen
Right
because
now? Okay.
yeah, because we before we going to the presentation, I would like to present you. And I will share
Okay.
my screen like this. Share screen. This one. Oh,
Wow.
you look good, 老师.
I don't know.
你很
No
好。
comment. No comment. I'm
You are
embarrassing, you
you
know?
are getting younger. It's been a decade, but you are younger than before.
Oh, really?
I will stop
Okay,
sharing.
when you go to Taiwan, I can treat you to the restaurant and and
Wait
provide you a
me.
lot of candy.
Wait me. I didn't pass the last ICDF scholarship, so I'm gonna try another.
Okay, no problem.
Just
And
bear
just
with me,
just
Laoshe.
waiting for you. Yeah, just waiting for you. Okay.
Just bear with me because you are my forever referee. I will ask a recommendation letter from you.
Yeah, it's okay. And I can prepare more candy for you
谢
because
谢
you are so
老师，
sweet.
谢谢你。I invite Gabby and Restu as well, but seems they don't join yet. You
Yeah,
remember
just
Restu,
depend.
right?
Yeah, just depends on your time. It's okay. I
Yeah,
just wait. Okay.
we can wait more than. You have meeting this noon in the department? Wait. Mas Elga, Mas Elga, you there?
Ya.
Mas Elga nanti bantu screenshot ya untuk dokumentasi. Barangkali nanti aku nggak sempat screenshot.
Oh, ya ya.
Nanti setelah Pak Imam ada nggak ya?
Sebentar, dicek dulu.
Ah,
Misalnya itu tadi pagi itu info Pak Imam tuh barengan dengan Monef ORHL.
oh gitu. Jadi ada yang mewakili Pak Fajar atau bagaimana opening remarks-nya?
Pak ya, biasanya sih PME atau gimana? Atau saya juga nggak apa-apa.
Ya, Pak Fajar juga nggak apa-apa, Pak. Selaku karyawarisnya.
Ya, biar segera dimulai aja ya Mas Elga.
Closure, we will wait for another 2 minutes.
No problem. No problem.
Okay, thank you. Should we start now, Pak Fajar?
Yes, please, Mbak Aini.
Okay. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. My name is Ar-Rahmatu Syafaqah Liaini, a junior researcher at the Research Center for Applied Botany, BRIN. It is my pleasure to serve as your MC and moderator for today's Botany Talk. I am delighted to welcome all of you, and I hope today's meeting will develop our understanding of how advancement in molecular plant immunology can be turned into novel and eco-friendly plant protection strategies. I would like to extend our warmest welcome to Bapak Fajaruddin Ahmad, Ph.D., the head of the research group. The head of the research group, bahasa Inggris,
Plant Genetic Resources.
Biogenetic Resources. Sorry, thank you, Pak Fajar, for Genetic of Plant Bioresources. And thank you, Pak Fajar, for joining us and for your continued support of this event. We are honored to have with us Professor Yi-Hsien Lin from the Department of Plant Medicine, National Pingtung University of Science and Technology, Taiwan. Thank you very much, Lao She, for accepting our invitation and for taking the time to share with us today. Before we begin today's scientific session, it is our honor to invite the head of the research group for genetics of plant bioresources, Bapak Fajaruddin Ahmad, Ph.D., to deliver his opening remarks. Kepada Pak Fajar, dipersilahkan.
Thank you, Bu Aini. Assalamualaikum warahmatullahi wabarakatuh. Good morning, especially distinguished speaker, Professor Yi-Hsien Lin, college, and also researcher, and maybe student, and maybe other participant from other institutes. So here, and on behalf of the Research Center for Applied Botany, National Research and Innovation Agency, or BRIN, I would like to extend a warm welcome to all of you to the Botany Talk series number 39. So today we are honored to host a lecture entitled The Botanical Shields Application of Beneficial Microorganisms in the Management of Plant Disease. I think this topic is highly relevant as sustainable agriculture to increase reliance on environmentally friendly approach to improve crop health and to reduce our dependence to chemical or pesticides. And beneficial microorganisms have emerged as powerful tool for enhancing plant resilience, managing the disease, and make I think it will be a great discussion, particularly will be a great discussion in this program. And it is our great pleasure to welcome our keynote speaker, Professor Yi-Hsien Lin, from the National Pingtung University of Science and Technology from Taiwan. I think Professor Lin has made a significant contribution to plant pathology and its application of beneficial microorganisms to in sustainable disease management. So we are grateful that he has accepted our invitation to share his experience, expertise, and I hope it can give some insights to us today. So we hope that this seminar will provide valuable scientific knowledge, also stimulates engaging discussions to enlarge our collaboration with international partners. So without further ado, it is great honor to invite Professor Yi-Hsien Lin to deliver today's keynote presentation. So please, Professor Lin, the time is yours.
Terima kasih Pak Fajar for your continuous encouragement and support for scientific collaboration and knowledge sharing. But before we proceed to today's presentation, we would like to take a group photo. Participants, if your camera is currently turned off and you are comfortable doing so, we would be grateful if you could turn it on and give us your brightest smile. Mas Adit, boleh minta tolong screenshot. Let's take the documentation starting from slide 1. Mas Adit, ready?
Siap, Mbak. Baik. 1, 2, 3. Slide kedua.
Slide
1,
2, 1, 2,
2,
3.
3. Slide 3, 1, 2, 3. And the last slide, 1, 2, 3. Thank you, Mbak.
Okay, thank you everyone. We have finished taking pictures. Now, let us move forward to the presentation. First thing first, I would like to present our speaker today. Let me share my screen first. Okay. Our speaker today is Professor Yi-Hsien Lin. He has obtained his Ph.D. in plant pathology at National Chongqing University, Taiwan, and is currently a professor at the Department of Plant Medicine, as well as the Dean of the College of Agriculture, National Pingtung University of Science and Technology, Taiwan. His research contributed to pathogen-associated molecular pattern-triggered immunity, mitogen-activated protein kinase signaling, induced resistance, and the utilization of Bacillus species as biocontrol agents. His work seamlessly integrates fundamental molecular biology with real-world agricultural application, helping in the development of sustainable alternatives to conventional disease management. Professor Lin has published extensively in leading international journals and received multiple distinguished awards for his contributions to plant pathology and agricultural biotechnology. Today, he will deliver a lecture entitled The Botanical Shield Application of Beneficial Microorganisms in the Management of Plant Diseases. I would like to encourage everyone to actively participate in the discussion. If you have any questions during Professor Lin's presentation, please feel free to post them in the chat box. Alternatively, after the presentation, you may use the raise hand feature if you would like to ask your question directly. We have prepared door prizes for 3 active participants. One participant will receive one of those 3 packages. So don't hesitate to ask questions and engage in today's discussion. We look forward to hearing your ideas. Okay, then I will stop sharing. Now, Lao She, whenever you are ready, you may share your screen and begin your presentation.
Okay, thank you. Can you see?
Yeah, Lao
Can you
She.
see the PPT?
Yeah, sure.
Okay, so thank you for the head of the Biotechnology Resource Center, and thanks for the introduction for Liani, my previous master student in Taiwan. And today I want to share something very easy for you to just express some application linked to the research and how to make the research link to the application about the beneficial microorganism. So I use the title today is the Botanical sheet, and also my title will be the application of beneficial microorganisms in the management of plant disease. Of course, we have some very short introduction about me, so I will not talk about me too much. Or I will just want to share you something. The most important thing today is I want to tell you about what is the plant immune system. And then we try to use the plant immune system to apply in the greenhouse or apply in the field, and how to apply in the field is just we can use the fermentation techniques. And the last part, I will introduce if we already have very good fermentation liquids of this kind of beneficial bacteria, and how to use the environment control to make the biocontrol efficacy become better. So this is the 3 part. So the first part, I will focus on what is the plant immune responses. This is the most important plant defense pathways, and we can use this kind of system to increase the plant health. So the first one, I will talk about what is the immune responses. As we know, the immune responses is always happen on all kinds of living cells. And why the living cells need the immune response? Because they are the most important thing is for their longer living. They need to face to a level adverse environment, such as the bacteria, viruses, or fungus, we so-called the pathogens. So the immune system is focused on pathogens. When we see on our human body, the immune system is also a part of the body. So it's the main role of the immune system. The most important key word here you can see is the recognize. So the recognize is the most important key word of the immune system. If we can use the immune system, and the immune system can recognize the pathogen, and our body will eliminate the pathogen, so we can keep our body very healthy. So the immune system is very important. And when we want to make the difference, we want to understand more on the immunity. we can separate into two categories. And one is the innate immunity, and the other is the adaptive immunity. What is the innate immunity here? Is a non-specific defense mechanism, indicates the recognition is not so specific. So in in normal system, I will show the figure later, and you can feel the non-specific defense mechanism is happen on the cell surface, and the cell surface can recognize all kinds of compounds or any kinds of molecules and to sense the pathogen or microbe is coming. So the non-specific defense mechanism is present throughout the entire lifespan of the organism. So this is the very basal immunity. Another is the we so-called the adaptive immunity. The adaptive immunity is when we face to a lot of years of COVID-19, we use a lot of kind of vaccine or some molecules of the COVID-19. When we use the partial sequence or partial protein as a elixir to trigger the specific immune response, so our body can face to the COVID-19. So this is so-called adaptive immunity. So the adaptive immunity is very specific and can be established. The defense system so appear to the expose the pathogen or by the vaccine. So we can understand the immune responses, and then we can know the immune responses have several following steps. The first one is recombination, activation, coagulation, and elimination and memory. So, but the most important thing I want to emphasize here is the recombination is the most important thing. Is indicates the cell need to know the pathogen. If our cell understand or recognize the pathogen, and we can trigger the further responses. So when we want to transfer our understanding from the innate immunity from animal to plant system, so we need to know we already know the animal immune system. And the animal immune system, the most important cell is our white blood. So and on the surface, on the membrane surface of the white blood, have some receptors. We so-called this kind of receptor is the pattern recognition receptors. And this kind of receptor have some function. And the receptor can recognize small molecules produced by pathogen. So we can see the keyword here, the recognize. So recognize if the cell can recognize the pathogen or the small molecules from the pathogen, and the defense can be triggered. And we can call this kind of small molecules as pathogen associated molecular patterns. We so-called the PAMP. So if I say PAMP in the first slide, you can know this is come from the pathogen, and this is a very small molecule patterns can be recognized by receptors. So when we we understand on the animal system, and when we transfer the pathogen become to the plant pathogen, when we transfer the cell to plant cell, when a plant cell can recognize the plant pathogen or the small molecule from the plant pathogen, we can so-call the plant immune system. This is very easy, the very basic understanding on the plant immune system. And when we want to know the plant system, we can just look at the picture. You can understand this is the plant cell, and on the surface of the plant cell, we can see this kind of structure. This is the plant receptor. The very common plant receptor we so-called the FLS2 is a kind of receptor can recognize the flagellum from bacteria. So we can see the PAMP when the flagellum or the PAMP release from plant pathogenic bacteria. And these kind of molecules can be recognized by the receptors on the cell surface. And this response we can so-call the PTI. Here we can see the PAMPs trigger immunity. And however, we can know another pathway is the plant pathogenic bacteria can use the type 3 secretion system to squeeze the effectors inside the plant cell. So we can see the effectors already penetrate inside the cell. And this kind of effectors can be recognized by the intracellular receptors. We can see the receptors here, and this kind of receptor can recognize the effector. This is so-called the effector trigger immunity. We can see here, and the effector trigger immunity in the textbook of plant pathology, we can so-called this is a kind of gene for gene theory, indicate a virulence gene from the plant pathogen. It can fetch to the resistant gene of the host. And this kind of gene-to-gene theory can bring out the hypersensitive responses. And this kind of hypersensitive response is also a kind of defense responses, or we so-called the resistant reaction of the plants. So when we combine the PTI or ETI together, these two pathways, one is from the cell surface and one is inside the cell. We combine them together is we so-called the plant immunity. And some important issue is about if the plant bacteria squeeze the effectors inside the cell, this kind of effector can also inhibit the basal defensive response of PTI. But all of the responses is just depends on the strength of the defense So the MAPK or ROS will control the strength of these defense responses. So this is a very easy figure to show you a lot about the PTI and ETI. Of course, we can see another issue here is the PGPR. Our study is want to find out a PGPR or if a bacteria can regulate the PTI or ETI, perhaps we can get a very new idea from this system to make the plant become healthy. But as you think, the PTI or ETI, which one will be easier to regulate? Of course, because PGPR cannot enter the plant cell. So the PGPR might have some beneficial effects on the PTI intensification. So the main role today is I want to introduce what kind of PGPR can intensify the PTI to make the plant resistant to a lot of different kinds of plant diseases. This is our issue, our story today. So what we want to use, what kind of beneficial microorganism we can use. We know the beneficial microorganism we so-called is all plant associate. And this kind of beneficial microorganism can help the plant and promote the plant growth. So, and also can induce the plant have some defense response can be triggered. Okay, and this we so-call this kind of bacteria, and is the plant growth promoting vital bacteria we so-called the PGPR. And and how about the use of this kind of PGPR? And in most of the cases, or in the world, we can know the bacillus or pseudomonas can be used for the PGPR, and these two species can two genus can help the plant and become or transfer plant become resistant to a lot of different kinds of um, crop and defense response to the defense different pathogen attack. And and the most important is the bacillus. Why we want to use bacillus? Of course, in I according to I remember in the Inis study, she also used the bacillus as a target. So why we want to use the bacillus? Because the bacillus species is one of the most common use for the microbial group among the PGPR, and a lot of a commercial product also use PG bacillus as a very important targets. Why? Because the bacillus species can bring very good biocontrol efficacy and in the management of wide range of plant diseases. And this kind of bacteria can also can promote the plant growth and in a lot of cases. And the most important thing is about the nature of the bacillus, because this kind of bacteria can grow under the aerobic or anaerobic conditions. Indicate if your oxygen is not enough, this kind of bacteria can grow still very well. So, but most important thing is for the shelf life, because if you produce the bacillus fermentation liquids, you want to sell to the farmers. The shelf life is very important. And why? Why the shelf life of bacillus is very good? Just because bacillus can produce the endospores. So when the bacillus use the nutrient from in the fermentation tank, and already and the sucrose or protein were exhausted, the bacillus can transfer itself become the endospore. So the endospore can help the bacillus have very high potential to develop the microbial agents. And of course, bacillus also have one very good nature. Just because the bacillus can produce endospore, can resist to the sunlight. So so when you apply the bacillus fermentation liquid or the bacillus cell into the field, the bacillus will not decline so rapidly in the field. So bacillus is a very good choice. So if we want to develop the fermentation or develop the microbial agent in the future, bacillus is a very good choice. Of course, when we want to use bacillus, we need to take care about some species of bacillus is not so safe. Including the Bacillus cereus is not a very good, is also pathogenic to humans. So we need to choose a safety Bacillus for the further study is more important. So this is the key. So just remember that point. And when we start from the research of the Bacillus, we know the Bacillus had a lot of different kinds of characters, including the bacillus have some antagonist activity or induced resistance or colonization was so good. So So the first one, when we want to use the bacillus for the biological control, a lot of people will do this kind of experiments. So you can see the picture here. You can see this is 3 different kinds of bacillus strain, PMB04, 05, or DR81. And we can see the PMB04 and DR81 have very good biocontrol potential because the antagonist activity of these 2 strains was so strong. You can see the clear zone here. You can see the clear zone here. However, the PMB05 is not so strong, just have very weak the inhibitory zone. And also, you can see this is focused on Ralstonia. Ralstonia is a kind of pathogen can cause the bacterial wilt. A lot of people already understand. And another is Asterovoras situ. So right now we so-called the para Asterovoras situ. Just depends on the name change. But we can see here the PMB04, 05, or DR81 have very good effects to and to have the antagonist activity to the Asterovoras situ. However, you can see here the DR81 have very strong antagonist activity. So we can raise a question here. You can understand. And the question is, if you want to apply this kind of strengths in practical, and which kind of strength would you choose? You can write down the answer in your notebook. But however, perhaps the answer is not so correct in my first presentation. And normally, a lot of people will choose PMB04 or DR81 as a very important target to develop the fermentation liquids or microbial agent, any kinds of microbial agent, because these 2 strains have very strong antagonist activity. And when we really inoculate or try to use this strain to control the plant disease, you can see the bacterial wilt for the control. All the plant were wilted, correct. And however, you can see the DR81, the wilting is still very severe. It's very similar to the control. And when we compare to the control, DR81 might be more severe than the control. However, in these 2 strains, PMB04 or PMB05, these 2 strains were very good, and PMB05 will be the best one to control the bacterial wilt. When we compare to the previous slide, we can see PMB05 to Ralstonia solanaceae, the pathogen of bacterial wilt, is not so strong, and PMB04 is strong. And when we compare the result here, PMB05 without very strong antagonist activity can bring the better biocontrol activity. Why? So raise a question here. So we think it might be only appear on the bacterial well. So we try to test another plant diseases. So we can see the bacterial fruit here. The PMB05 will also bring various superior biocontrol efficacy than the DR81. You can see the DR81 still have a lot of symptoms. So when we transfer to this true for bacterial disease, when we transfer to control the fungal disease, we can see this is the anthracnose on strawberry. The anthracnose on strawberry is a very important disease in Taiwan. So when we use the pathogen to inoculate the strawberry, we can see the strawberry will show severe wilting. And we can see the PMB05 not only reduce the occurrence of the anthracnose, but also improve the plant growth and then to make the strawberry grow very well. And PMB04 also have very similar function, but PMB04 also have some plant wilting. When we transfer to the citrus canker, we can also see the PMB05 have very good control efficacy to the citrus canker, and also have very good control efficacy to the bacterial leaf spot on rice. You can see the control efficacy is so good, and also have very good control efficacy on Oncidium orchids. So we can see after the treatment of PMB05, the co-inoculation of pathogen, you can just only see one spot on the leaf, no very severe several symptoms. So we can know this kind of disease control of beneficial microbe on plant is not only correlated with the strength of their in vitro antagonist activity. So the in vitro antagonist activity, according to the result, seems not so important, right? So we want to understand what kind of possible mechanism involved in the disease control. So we just try to think about the mechanism again. One is the antagonism. Antagonism indicates the antagonistic activity we already see. It might be not so important. Another is the induced resistance. So we want to talk about the induced resistance a little bit in the future. And also another is the colonization. But the colonization is all about the biofilm formation on the rhizosphere. So I just want to show something about the induced resistance, because induced resistance have very, very good effect. And induced resistance also include the plant immunity intensification. So I want to talk about the issue in the first part. And then let's see a case firstly. And we know the bacterial fruit blotch of watermelon is a very important or very severe disease of the watermelon. And of course, you can see the symptom on the watermelon and also the melon and on the leaves. And this is a kind of seed-borne pathogen. So when we apply PMB05 on seeds or on the plants, what will happen? So we can see the case. This is the very similar case when we apply the PMB0405 and DR81, and we can see after 48 hours inoculation, co-inoculation with the pathogen, we can see the co-treatment of the PMB04 or 05 or 81 with the pathogen. We can see after the co-treatment of the pathogen and the beneficial microbes, you can see only PMB04 and 05 can have very good control efficacy to compare to the control, and also have very good better control efficacy than DR81. And of course, we can know here that DR81 with very strong antagonist activity also have some biocontrol efficacy to when we compare to the control. But the control efficacy is not so good, not so good as PMB04 or 05. So we will ask a lot of questions. What happened? So after few years study, we want to focus a lot on about the plant immunity. But how to assay the plant immunity? Right now, I can explain a little bit to you. And we can see the panel A or panel B. And panel A, you can see the green color. If you can see the green color, indicate this is use a fluorescent dye to stain the reactive oxygen species. If you can see the more green fluorescent dye produced indicate that more ROS can be produced in the cell or in the tissue. So we can just see the green color. And another important hallmark of the PTI, we so-called the PAMP trigger immunity indicator, is not only the ROS, also the Kalos deposition. So when we see the panel B under the blue background, you can see the bright blue points. These bright blue points indicate the Kalos deposition. And if you can see more Kalos deposition, indicate the PTI triggering is more activated. So we can use the ROS generation and the Kalos deposition to see what we call inoculate the pathogen and the pathogen and the beneficial bacteria. We can see PMB05 have very good effects on the fluorescent dye activation including the ROS generation and the Kalos deposition. However, you can see here the DR81 will decrease the plant immune signals. You can see the blue dye will produce just very seldom, and also you cannot see a lot of blue light points can be produced in the tissue. So under this kind of experiment, we can confirm that perhaps PMB05 can intensify the plant immunity, especially focused on pampa trigger immunity. However, the DR81 may reduce the PTI. So this is two different kinds of effects on the plants, especially on the watermelon. So when you first apply the PMB05, PMB04, or DR81 on the seeds, you can see the PMB04 and 05 have very good effects to control the bacterial fruit blotch on watermelon. When we use the seed application, we just soak the seeds in the culture of these three strains, and then we apply the pathogen, and we can know the PMB04 and 05 can have very good control efficacy. Of course, DR81 might have some better effect than only the pathogen control, but you can see the control efficacy is not so good. So after that, we can try to emphasize something. We can go back to the PTI, understanding on our PTI. You know, this is a kind of bacterial pathogen, and the bacterial pathogen can produce a PAM to trigger the PTI. And of course, the pathogen can produce an effector to trigger ETI. But our rhinobacteria here, rhinobacteria, the PGPR, if our PGPR here can enhance the PTI responses, well, some just a little bit inhibit the pathogen, might help the plant become very easy to become resistant, will make the plant very easy to become resistant to the disease. So how about this kind of, if the bacteria can have some function on the PTI intensification, we can have some advantages, because a lot of people already report some PGPR can trigger the induced responses directly. But however, you can see the example here, the PMB05 will not intensify will not induce the resistance directly. It only intensifies the PTI. So the advantage will be no energy consumption, and this kind of resistance might be very poor spatial. So this is the PDI intensification, the character of PDI intensification bacteria have. So we can use this kind of bacteria. So I can just want to emphasize here, you can know which type of beneficial microbial strain can be preferable. And one is the priming strain. Priming strain is indicate the microbial strain can direct induce disease resistance or the pathways, such as the SA pathway or osmotic acid pathway. And this kind of bacterial strain is the Bacillus belenenses FZB42. This is have a lot of published paper already on the website. And another is very new bacterial strain is about the immunity intensification. This kind of microbe strain is just depends on the plant immune system. So we can know when the plants recognize the pathogens, and this kind of microbe can further intensify this kind of response. The most important example is the Bacillus amyloliquefaciens PMB05. So in the first story, I will talk about more on PMB05, the most important bacterial strain we found in our lab, and also we got the United States patents and also the Taiwan patent, and this is a very important strain. So I want to share something about this strain to you. So the second step is I want to say something about how to use the PMB05 for the fermentation formulas. The fermentation formulas, we already have some fermentation tank in Taiwan just for the education, not for the commercial produce, just for education for all the students. All the students can use the fermentation tank in the classroom, and then they can use for their study. So this is some very good equipment for the students. So we just use this kind of biofermentation tank for the student for the field application. And fermentation techniques, of course, we know is use very seldom bacillus and use this kind of fermentation tank and to produce a lot of bacillus. And we can apply in the field. And this kind of fermentation can be achieved in a very short time. How about the fermentation? We can start from the 500 milliliter of flask to 10 liter and to 30 liter. So in the lab, we just use 500 milliliter of flask and go to the 30 liter fermenter directly. So right now I will show you some very important information about the fermentation. If you want to do something good in fermentation, you need to prepare the growth curve of bacillus or of your target bacillus or target bacteria first. And this is the growth curve of bacillus. We can know that after our inoculation into the medium, we can use the LB medium. The LB medium, after we inoculation the bacillus into the medium, we can see under it, when post-inoculation, after 4 hours to 6 hours were under the log phase. The log phase bacteria is a very important initial inoculant into the fermenter. So we need to use the source of the bacteria in these time points. And after we use the bacterial source in these time points, we can use the fermenter. But the most important key is how to use the fermenter. If we purchase a new fermenter, but the formulation is very important. How to make the fermentation formula. Can you ask the chat GTP? It's not easy because the chat cannot tell you the correct answer. So the most important thing is just come from a basal medium from the lab. If you have, after the seminar today, you will know a little bit about how about to start up the fermentation, because I will show you the formula. And we can see the formula here. We can use the 1:1, 1:2, 1:3. This is very difficult for you to understand. And you can know this, the number 1 here is indicate 1%. 1% of carbon and 1% of nitrogen. So we need to provide the carbon and nitrogen sources. And these two sources were the basic. And so if we use 1% carbon and 1% nitrogen, we will produce the cell reach to the 10 top power to 10 to the power of 8. So we can see after we transfer, we try to test a lot of different kinds of formula. We can see the 3% of carbon and 1% of nitrogen will be reached to 10 to the power of 9, and the sporation will reach 85%. Indicate 3% carbon and 1% nitrogen will be the best one in our test. And this is case by case. And if you want to use any kinds of bacterial strain, you need to test the formula first. And the most important thing is the 1%, 1% and the 1% here. And you can see the totally median is recommendation not over 5%. Indicate in 500 milliliter of medium, you just have the totally number of the sugar or the protein you added cannot over 25 grams of these compounds. So what is the carbon source or nitrogen source? The carbon source is the brown sugar. So you can use the brown sugar as your carbon source. And you can use the yeast powder as your nitrogen source. So if you can combine these two compounds, and it's enough. And sometimes a lot of people don't want to use yeast powder as a nitrogen source. They can also use the soybean powder. The soybean powder can be also used as a nitrogen source. So after this kind of test, you can know when you go to 10 liter and 3% and 3% of carbon and 1% of nitrogen will be the best one. And you can see the picture here. This is a kind of yeast extract and yeast powder, and this is the brown sugar. And the most important thing is we need to check the shelf life of fermentation broth after we produce a fermentation broth. You can use an assay, very easy. You just put your fermentation broth in 54 degrees Celsius and for 40 days. And if you can see the population will not have very strong decreases. Anyone have any problem?
Yeah, wait, Loza. I'm trying to mute the participants.
Okay, so continue.
Yeah.
Okay.
Sure, you
Okay.
can go on.
Okay. So you can see after 40 days, you can see already still have over 95% of survival cells. So indicate after this kind of work, we can guarantee this kind of fermentation broths might have around 2 years of the shelf life. However, we can is if we only use this kind of experiment to demonstrate the fermentation broth have 2-year shelf life is so sometimes some very dangerous. So in the lab, we also need to check that according to the time and the temperature, we can see when we put in the 4 degree, 25 degree, and 37 degree, we can see when you put in the room temperature or in the refrigerator, will make the PMB05 fermentation broth more stable. You can see after 12 months, it still have over 90% of survival cells. This is a very important step. If you after you get the fermentation broth, you need to use this kind of test. And after we got the fermentation broth or liquids, we need to test in the field what kind of targets is the best we can test in the field. Is about the citrus or lemon. We can you can see here, this is the bacterial canker and happen on the lemon, and it's also a very important disease, citrus disease in Taiwan or in America. So we use these kind of fermentation liquids to try to control this kind of disease. And of course, we need to test the how about the biocontrol or the plant immunity regulation in about the fermentation liquid. We can see when we use the bacterial suspension directly, you can compare to the blank, we can see the green fluorescent produced indicate the ROS can be expressed. When we use the PMBFL2A indicate that we use the fermentation liquid, and the fermentation liquid is 204 dilution. We can see, very strong green fluorescent dye can produce, indicate the ROS can be elevated. And also, we can see after we use the PMBFL2A, we can also see very strong catalyst deposition will also produce on the lemon leaves. The most important thing is this test is carried on the lemon. Okay, so this is about the plant immunity part. And when we actually inoculate on the plants, we can see the disease will reduce very strongly. And also, the field chill is very important. After we apply the PMBFL in the field, you can see the disease severity will decrease after 3 months. We just applied twice. Just applied twice on the plants. We can see under the normal situation, we did not use any kind of treatment. We can see the population or the disease severity in the field is go up or go down, and we can see the PMBFL treatment is only go down. So the disease after 3 months is almost disappear in the field. This is a very important demonstration. We get a success in the field to demonstrate the fermentation liquid can successfully control the bacterial canker on the lemon tree. And how about the dosage forms or fermentation products? When we get the fermentation liquid, is enough? Not enough. I want to apply the fermentation products such as seeds. So we want to we can control a lot of different kinds of diseases. The cabbage can be applied because the cabbage have this kind of disease. It's also very severe in the field, especially worldwide or in Taiwan. And when we want to control the black block disease, and we try to use this kind of machine. This kind of machine is a kind of air dryer, which we can so-called the spray dryer. The spray dryer is try to make the fermentation liquid become powder. So we can use the powder to mix with the seeds. After the mixing of seeds, we can get a very good control effects. So this is just the process. I don't want to focus a lot on process. I just want to show you, and this is the number here is indicate the temperature. If the higher temperature can, after you can see the injector here. The injector have higher temperature, and the spray will produce more powder, and the conversion ratio will be higher. So the higher temperature will make the bacillus can have more powder producing, and also the high temperature will reduce a lot of other environmental bacteria contamination. So this is a very good points or way to produce the powder. And after we produce the powder, we can see the powder can very good effects and will not affect a lot of generation rates when we use the very high temperature to 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 make the powder. When we produce the powder and mix with the seeds, and we compare the nature of the germination, we can see the germination will not affect by the powder. And after the most important thing is you can see this is the powder mixed with the seeds. And this kind of method can let the PM, you can see the PMBO5 can control the bacterial growth very well. So the powder is a very good material to and for the farmer can use the seeds directly. However, a lot of people will ask, this is perhaps this is not the best method, correct? And this year we also published a paper on plant disease. We used a kind of compound such as the agenate. Agenate is a kind from the seaweeds, and this kind of agenate will make the powder attach on the seeds, and we will not see a lot of powder on the seeds. We just see the seeds have some coating like the color on the seeds, and this kind of seeds when we apply on the watermelon, we can have very good control efficacy to control the the bacterial fruit blotch of watermelon. And this is just a case to show you when we use this kind of powder to mix with the seeds is a kind of method to control the bacterial growth. And the last part, I want to focus a little bit on the nutrient source or plant immunity or the environment. And we know plants have a lot of nutrient requirements, including the macronutrients or micronutrients, and which kind of nutrient can be applied to increase the plant immunity of PMB05. I just want to ask a question. So I use the calcium. We know that when you apply the calcium will increase, such as the PMB05 added another the calcium carbonates will increase the HR, indicate the calcium carbonate will help the PMB05 have more function on the plant immunity intensification. So after we say on ROS, you can see here, we can just see here is okay. PMB05 and calcium carbonate will have more ROS generation and more catalyst deposition. And this kind of effects will bring the PMB05 with the calcium carbonate have better control efficacy on the bacterial wealth. So the nutrient is also very important. So the calcium carbonate can be applied in the field. If a lot of people don't want to use or cannot use PMB05, if they can apply the calcium carbonate in the field, also have have some effects to increase the plant disease resistance. This is a common example here. And also, here is a demonstration. When we want to use, when we know the calcium carbonate is very important, and the calcium is also very important to plant immunity. So we want to demonstrate something. So we use the lanthanum chloride. And lanthanum chloride is a kind of calcium channel blocker. When you use the blocker, we can see the lesion of symptom will increase, indicates when we block the calcium channel of the plants, the PMB05 will reduce its control efficacy. So the calcium is very important. When we added some calcium into the powder, when we combine the calcium into the powder, and we can see the powder after we apply the calcium, when we compare to the SD160 is without calcium. All of the application of calcium will increase the control efficacy to the bacterial rot. So it's a kind of strategy when we want to make the PMB05 have more or better effects on plant immunity intensification. We can use this kind of strategy, and 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 the antagonic activity, or when we apply the calcium, will increase the antagonic activity? No. So we can skip the data. So, but most important thing is in the field. So we grow. We try to apply in the field. We can see the application of powders and also the fermentation liquid together will bring the best effects in the field. And also we can see the production of the cabbage will become bigger, and the weight will increase over 30%. It's a very good effect on the plant growth and also can control the disease. And this part, I will focus on the light conditions. And the light condition is very important when we know a lot of people want to apply the PMB05 or want to apply the microbead in the field. But the environment is so important. So this topic, I will just talk a little bit on how to make the PMB05 have very good plant immunity intensification, or it just depends on light. So we use different kinds of light intensity, and we see the 50, 100, and 200 indicate the strength of the light intensity. You can see the higher light intensity will make the disease reduce, and PMB05 under very higher in light intensity, above 100 or 200 light intensity, will have very good effect to control the bacterial wealth. But the PMB05 under lower light intensity, you can see we apply the PMB05 can increase the plant immunity. However, under low light, no effects. So the PMB05 cannot successfully control bacterial wealth under low light. So this is a very important concept or idea. In the future, if we want to use the beneficial microbead, we need to take care about the environment, especially on light. And the plant immunity, the energy source of plant immunity is come from the sunlight, and of course, come from the photosynthetic system. So this is a very important finding we can see. And we can see the 8-second videos indicate in the beginning, we can see under the low light without PMB05. I make the video from the AI. You can see just 8 seconds. And when we transfer to the highlight with a lot of shiny PMB05, will make the tomatoes grow very well without bacterial will occur. So this is also very important concept. We can know not only the beneficial microbe, not only the pre-immunity intensification microbe, we need to cooperate with the environment. The environment is also very important issue. Okay, and in the future, I want to say something about the future. We need to have a lot of improve indicate if we have a lot of light, CO2 or humidity or temperature database. When we try to add this, input this data into AI, and in the future, we will have a very good helper for the outputs. And the helper will, or the output will help us to understand what kind of situation will be the best way to use the beneficial microorganisms. And we in the NPUST, we want to develop this kind of system to make in the future, if the farmer get the APP, if the farmer grow the tomato, he just need to click the tomato. All of the environment is ready. And then if he can apply the beneficial microbes, will let the farmers, they don't need to use any fungicides or chemical fungicide or any chemical pesticides. They just need to use the microbial agent in the field, including the seeds or spray the fermentation liquid, and cooperates the environment. The disease will not happen, and they also have very good harvest in the field. So this is our goal in the future. So in NPUSD, we try our best to build up the database. And the final, I want to say, well, if we have microbe, we can use the antagonism, induce resistance and colonization. More important is we can use the nitrogen source for the cooperation and life. And thank you for all. I can go back to the to stop sharing. Hello?
Yeah, Lasha.
Yeah. Can you understand?
Oh, that's a very interesting presentation. And right now in Indonesia, there is national program called Free Nutritious Meals, Makan Bergizi Gratis. So they usually just put add simple fruits like citrus or banana to complete the meal tray. So the the production of citrus in Indonesia is quite something right now because of this program. So I think this how to say, these approaches can be used in Indonesia as well to maintain the plant production.
On
Okay.
citrus?
Yep.
On citrus?
Yeah,
For
citrus
banana?
oranges.
Oh, so you
Yeah.
can try.
There I think canker citrus is also a big thing in citrus production in Indonesia,
Okay.
just as you explained. So, before we go into the Q&A session, I'd like to remind the participants to fill the attendance form that have been shared in the chat box. Well, sure. I hope you don't mind that I will summarize your presentation in Indonesian, Keima.
Yeah,
I will summarize your presentation in Indonesian, Keima.
of course.
Oke, then. Thank you, Langsung. Oke, Bapak Ibu semuanya, dari presentasi Profesor Lin tadi, kita mempelajari bahwa tanaman bukanlah organisme yang pasif ketika dia terinfeksi patogen. Tanaman juga memiliki sistem imun alami yang dapat diperkuat melalui aplikasi beneficial microbes. Pendekatan ini menjadi perspektif baru dalam pengendalian penyakit tanaman yang tidak hanya menekankan pada upaya membunuh patogen, tapi juga meningkatkan kemampuan tanaman untuk melindungi dirinya sendiri. Jadi dengan pengaplikasian beneficial microbes tadi, tanaman bisa memberi alarm pada dirinya, bahwa there is a danger, ada bahaya di situ. Penelitian ini relevan dengan ruang lingkup riset kami di Pusat Riset Botani Terapan, yakni Fundamental Plant Biology melalui pemahaman molecular mechanism sistem imun tanaman, pengembangan plant product melalui inovasi biocontrol berbasis mikroba, serta pemanfaatan plant genetic resources melalui pengembangan tanaman yang lebih tahan penyakit. Thus, Professor Lin's research demonstrates how basic research on plant microbe interactions can be transformed into practical solutions to promote sustainable and eco-friendly plant production. Now, we are going to the Q&A session. We have several questions here. Let's talk. The first one. The first one coming from Bapak Muhdar Davis. Bapak Muhdar Davis, you there?
Iya,
Ya, Bapak dari mana, Bapak?
dari Botani Terapan.
Oh ya, mohon maaf. Terima kasih pertanyaannya, Bapak. Okay. So also there is some
So
question.
I can answer the question,
Davis.
right?
Yeah.
So I can
Yes,
answer the
just
question.
Yeah, you can directly answer the question.
Okay, and the first one is PMB05 have been proven by just PMB05 can intensify the brain immunity. However, but PMB05 cannot have any compound can trigger the defense system directly. And the PMB05 can intensify the immunity indicates the defense response only can be activated when the pathogen exists on the cell surface. If the pathogen is not exist, the PMB05 will not have any function on the defense responses. Can you understand? Okay, well, I can answer the question first, and then if you have further question, I can answer. And the second one is very similar, right? And what is the active compound? I don't know. I still need to try to find out the answer. But the question number 3 is a very good question. The PMB05 will not have any function on SA or JA pathway. And if the pathogen can trigger the SA pathway, the PMB05 can help the SA pathway more activated. But PMB05 directly cannot have any function on SA or JA pathway. Okay? And the next, I don't know if
Another
I answered.
one. This one from Pak Rudianto.
Oh, it's a very good question. The farmers use a lot of different kinds of chemical pesticides, right? And of course we have some study, and if any kinds of microbial strain want to apply in Indonesia, we are also recommend to do some research about how about the cooperation with the beneficial microbes and the fungicides. And of course, the PMB05, we also have this kind of test. We try to use a lot of fungicides or insecticides or herbicides, and PMB05 were some and most of them were resistant to the pesticides. But however, some fungicides, such as the copper, the PMB05 will not show very high resistance. It's sensitive a little bit to the copper fungicide. So under this kind of situation, we cannot use copper fungicide and cooperate with the PMB05 microbial agent directly. So we can choose another fungicide to use in the field. This is my recommendation. And the number of, yeah, the very similar
This
issue.
one from Nurfianto Giar Pangidung. I think
Yeah,
it's similar to the previous one.
a lot of words. Uh,
existing beneficial
and the question and right now I cannot have a very good answer to answer the question, because the microbes, of course, the root microbiome will be affected by the different cultivars of plants, and the issue about the microbiome of PMB05, I cannot answer that because I did not have this kind of research yet. So I have some cooperation with some new faculty in our school, and perhaps the professors will help me to answer more questions in the future. But right now I cannot talk about a lot of how to do to make a precisely design of beneficial microbiome. It's a very difficult question for me. I'm sorry, right now I cannot answer you. And the multi-pollination based cycle, yeah. Oh, uh, how about the future of the fermentation techniques? Just like what I say in the PPT, and in the future is the fermentation technique is not so hard. So a lot of people can use this kind of formula to produce a lot of fermentation liquids. But most important thing in the future is just try to find out the additive. Just indicates if you a lot of people can understand the fermentation liquid, but you have another other extra solution B. When the farmer want to use the fermentation liquid as a solution A, and they need to mix the solution A and B together, can bring the best function on disease control. So the secret is happen on the solution B. And today I also share something about the solution B is you can use the calcium carbonate. Calcium carbonate is also one kind of solution B. So the technique is have perhaps in the future we'll have a lot of secrets, and the secret is just depends on the formula. So 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 it's also case by case. A lot of very good strain can be found in Indonesia, and the system can be also applied in Indonesia. And a lot of company will have a lot of function on the material, so they can use try to find out the solution and will be better. Okay, I don't know. I can if I answered the question.
I remember my master's study back then. We assay the number of bacillus 1 month after application. Despite we were applying just on the seed, we
Yeah.
found the bacillus moving and grow in the stems and in the leaves. But this is the question number one from Pak Nurfianto. But unfortunately, we didn't assay the effect of pesticide toward the bacillus development back then. So maybe next time, Lao Sư.
Yeah. I also have some result about the pesticide application and combine the pesticide application and to control the disease. And of course, I think this is case by case. Different kinds of bacillus strain need to test its effects to the pesticides. So after the testing, you can further apply in the field. So I don't know the PMP05 case is also correct or very similar to other bacillus strains. So it need to be tested in the future. Okay.
Okay, we have someone raising her hand here. Bu Liliana Baskorawati, would you like to ask your question directly? Ibu Liliana.
Okay, thank you very much, Mbak Liani, and thank you very much, Professor Yi-Hsien Lin, for your presentation. So, because my background is from forestry, actually, so I would like ask about the general information. Like you mentioned in your presentation that plants to have an immune response. We also know that a plants resistant to disease is inherited from its parents. So there is that plant that resistance is usually is inherited from the parents. So can we actually tell that from the plant's immune system also this is inherited from the parents? And how about the forestry trees that usually hardwood, we have hardwood and long life cycle. Is the adaptive immune response what plays in this biggest role also in the disease resistance? Thank you.
Thank you. The Anna?
Yes,
Perhaps
Lily.
Lily. Thank you, Lily. And you asked a very good question about how about the balance of the forestry and the how to apply our microbes, right? And I think it is very difficult if you want to use the bacillus, and this bacillus can regulate the brain immunity, and the balance would be happen on the energy of the photosynthetic system, right? And photosynthetic system, the total energy will apply for the one is for the harvest, one is for the disease control, and how many percentage you will use for the disease control, and how many percentage you will use for the crop production is a very important issue. So if we use a microbe with so-called the priming effects, the priming effects indicate we use the microbe and the plant will induce the resistance. will use a lot of energy for the disease control. So for the total energy, the production will be reduced. Okay, and if we use the PMB05, this is the plant immunity intensification, indicates the plants need to fast through the pathogen. When the plant fast through the pathogen, will trigger the basal response. And PMB05 have an effect to intensify the effects or the responses. And this kind of disease defense responses is only occur when the pathogen coming. And when the pathogen is not coming, the PMB05 will intensify the plant growth. So the plant can obtain more photosynthesis energies. So it will help plant have more production. This is why I understand the balance. I think I cannot answer you a lot of, because I did not have a lot of idea on this mechanism, but I just according to what I see in our actually situation, we can see this kind of formula. And how about another question is how about the balance in the forestry? However, I did not and in our school we have some faculty about the forestry, and also they use a lot more different kinds of microorganisms to apply in the forestry. And but I never have this kind of experience, so I only focus in the fields. So so I'm so sorry I cannot answer you the idea about the link to the forestry. Okay.
Okay,
It
that's
is
all
okay.
right. Thank you very much.
Thank you.
Okay, is there any other questions? Oh, we found another one from Nurilha. Ibu Nurilha, you there? Oh, wait. This one, Losho.
Yeah.
This
Oh,
one. The last questions from
yeah.
Nuril.
Yeah, the endophytic microbes is very good and for the application. And just like the Lily's question, and Lily also in a lot of application in the forestry, they use the endophytic fungus to increase the plant growth. And of course, and how about the endophytic microbes? Bacillus is also one kind of endophytic microbes. And in our study, we also demonstrate PMB05 can be endophytic growth inside the plant tissue. So when the bacillus exists in the plants, of course it can attack the plant resistance. And just like the previous question, and the plant resistance increase is to have two ways. One is the primary effect, one is the immunity intensification. And these two ways were possible. And these two ways we only need to care is if we use a microbe can improve the plant growth. The energy, we saw the balance of energy. If the microbe can improve the plant growth, will not have a lot of effects on the production. But if the bacillus strain or the microbial strain cannot have any effect on plant growth promoting, and is not so good to recommend for the farmer to, because we need to have the basic plant promotion, plant growth promotion, and then they can see the advantage of they also have the function on disease control. And if we can combine these two functions, and it would be more easier to promote for the farmer side. So I think if we can combine a lot of different functions of the microbes, it would be more practical or more economical for the farmers. This is my answer. Okay.
Okay, is there any other question? Oh, if there is no question anymore, there is question for me, Lausche.
Okay, no problem.
This one, I typed it in the slides. Wait. This one. I'm particularly interested in the molecular mechanisms underlying bacillus-mediated disease resistance. So if we want to know which genes are activated during pathogen infection following bacillus treatment, should we start with targeted analysis of well-established defense marker genes or use like the transcriptome-wide approaches like RNA sequencing to identify novel regulatory pathways?
Yeah, I can answer you the question first. The first question is depends on your budget. If you have enough budget, of course, the RNA sequencing, especially focus on the messenger RNA sequencing, is recommended because you can know a lot of new gene were expressed, downregulate or upregulate. But the time point is very important. If you want to focus on in the SA pathway, at the SA pathway you can assay the plant messenger RNA after 24 hours. But if you want to assay the early responses or PDI activation, you need to assay the gene expression after your inoculation in 1 or 2 hours. So it just depends on the type. And if you have enough budget, of course, RNA sequencing is recommended. But if you just want to see the expression or you want to use some marker gene, the marker gene on the PDI, just like the FRK1 gene, or the SA pathway, you can assay the PR gene, or the J pathway, you can use assay the PDF 1.2 gene. They are all available for your research. But if you want to get more new ideas from the topic, messenger RNA sequencing is recommended. Okay, if you can find more budget. But if you want to do it in Taiwan, it's still available. Okay.
Wish me luck, Lausche, wish me luck.
Okay.
Another one, Lausche. Your research
Okay.
highlighted the importance of ROS burst in bacillus-mediated immune priming. Given that ROS are known to regulate the production of several secondary metabolites, do you think phytochemical profiling could serve as an additional indicator of immunological activation?
And the answer is the ROS. We will not see ROS as secondary metabolites. ROS is a kind of free radical. It can produce very rapidly in 1 hour. So it's just the electron. After the plant immunity happen, the electron will transfer to the oxygen. The electron transfer to the oxygen will produce ROS. ROS can be a signal, and the signal can be activate the first MAPK pathway, and also the SA pathway. The first one is the MAPK pathway. After the MAPK pathway activate, the MAPK pathway will first activate the SA pathway for the such as NPR1 and also the PR protein activation. So I think the RS is just as a signal. So this kind of signal can be regarded as a very key signal for our observation in the cell to see the brain immunity is occurred or not. So it's just a signal. So it might be an indicator, but I don't know. I don't know if the hours and chaos were the indicator, but I don't know have other immunological indicator or I don't know. We just use these two signals for the analysis. Of course, if you want to assay the defense response, in the last part, you can also see the hypersensitivity response is occur or not. It's also a way you can do. Have you ever investigated combined metabolites and JavaScript? Yeah. I already have some researches on. I also have some study on PNB05. I try to see the DNA sequence of PNB05. Try to compare other bacillus strain to find out a new gene, but I am not successful in right now because I cannot have enough people to analyze the data. And how about the PDI marker gene is about the gene on plants. Of course, I already have some result on messenger RNA sequencing or after I apply the PNB05 or after the core application of pathogen elicitor. And right now I cannot answer you I cannot provide you a lot of answer because I did not finish the research yet. So we have some cooperation with the teacher in our department, but we need to have more discussion about this part. So right now I cannot answer you the question. Okay.
So it sparks an idea for my future research to combine the phytochemistry and beneficial microbes in plant protection strategies. 谢谢老师。
Okay. Perhaps when you come to Taiwan, we can have more discussion. Hey, I will present my candy for you.
I have a son, 老师. It's not easy for me to go. It's not easy.
Okay. Just wait, okay?
谢谢老师，谢谢. Is
No,
there a
very good
question? I see Bu Gaby and Bu Restu here. Would you like to turn your camera on and say hi to the 老师? Ibu Gaby, mana tadi ya?
Maybe, perhaps not here. I
Oh,
remember Gaby. Oh,
yes.
okay.
She's leaving the meeting room. She's going to Birmingham, 老师. You know that?
Oh, yeah, I know.
I'm so proud.
I'm
So,
so proud of you. Me too.
谢谢老师. I do nothing.
No. You can understand the research is just step by step, right?
Not here, 老师. There are a lot of people.
I cannot talk too much. I'm sorry.
Baik, Bapak Ibu, apakah masih ada pertanyaan? Jika tidak, kami menunggu panitia untuk mengambil 3 nama. We will choose 3 participants. 3 participants to get the prizes. Thank you, 老师。老师, I just sent the in a private chat here. Would you like to check it first? You can just answer in the chat room, in the private chat room. K, Ibu K.
Not too much of words. The words are so small, so I cannot see the words so clearly.
No. Oh, this one. Oh, I think you don't find my chat. Several participants have asked whether they could have a copy of your presentation slides. Would you be
Of course.
comfortable with
course.
us? Okay, then I
Yeah,
will share.
No problem.
Thank you,
No
Laosha.
worry about it. Okay.
Thank you, Laosha, for your generosity. So we have 3 names here to wait. Okay, we have 3 names here. Ibu Liliana, Pak Muhdar, dan Pak Nurfianto.
Thank
They
you.
will. Terima kasih Ibu, selamat. Terima kasih sudah mengikuti.
Can I choose 50 disposable battery dish?
Sure. Boleh Ibu, boleh. Nanti kami kirim. Ibu posisi di mana?
Jogja.
Oh iya, nanti kami kirim ya Bu. Nanti saya hubungi Ibu.
Siap, terima kasih.
Iya, baik Ibu. Terima kasih. Okay, there is Pak Muhdar dan Pak Nurfianto. Are you guys still here? Oh, I think they left the meeting. Okay. As we come to the end of today's botany talk, wait. I would like to express our sincere gratitude to Professor Yi-Hsien Lin for delivering such an insightful and inspiring lecture. 老师，谢谢你 for your inspiring presentation and for sharing your remarkable research with us today. Your research has deepened our understanding of how molecular plant immunity and beneficial microbes can contribute to sustainable agriculture. Personally, I found today's lecture especially inspiring as my own research interests lie in plant microbe interactions and molecular mechanisms of disease resistance. On behalf of the organizing committee and Research Center for Applied Botany, BRIN, we sincerely hope that today's event will be the beginning of future scientific exchanges. We look forward to collaboration opportunities with you and your research group in the near future. Thank you once again, Laoshe, and thank you everyone for joining us today. We wish you all a wonderful day, and we look forward to seeing you at our next Botany Talk. Terima kasih semuanya. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, everyone.
Assalamualaikum. Thank you.
And
Yes,
thank you,
Laoshe.
Lily. Thank you, Lily.
Thank you.
Also, we are not going to hold you no more. So if you
So I can leave the room.
yeah, you can leave the Zoom.
Okay.
So I will send the certificate through email.
Yeah, it's okay. Hope I can see you soon.
I hope 老师，谢谢。
Okay.
Just bear
Bye.
with me, because you are my forever referee.
Okay.
I will
Thank
ask
you all.
another recommendation letter, recommendation letter.
Yeah, just if you need, just find me, okay?
谢谢老师，
You're
再
welcome.
见，
And thank you all the friends from Indonesia. Thank you. Bye bye.
bye bye. Terima kasih semuanya. Mas Elga, Mas Adit, ini aku end meeting for all atau bagaimana? Oh, Bapak Ibu, barangkali ada yang belum mengisi link sertifikat, boleh diisi dulu sebelum meninggalkan Zoom. Eh, Tadi Bu Liliana, Pak Muhdar sama Pak Nurfianto. Pak Nurfianto, Botani Terapan bukan ya? Mas El, Mas Elga, you still here? Ya, gimana nih menghubungi orang yang bukan boter? Hmm. I will just reach him through the email.